Hello, and welcome to the first of the 2024 Gairdner Global Perspective Panels presented by TELUS Health. I'm Sarah Devonshire, Director of Awards and Outreach at the Gairdner Foundation, and it's my pleasure to welcome you all here today. Over the coming months, we will be hosting a series of global perspective panels led by our early career investigator competition winners from recent years. And together with presentations from world-leading researchers, they will be sharing the latest developments in precision cancer care and omics, prospects for mRNA therapeutics, the microbiome from the microscopic to the global, and AI in healthcare. We hope you'll enjoy today's discussion and visit Gairdner.org to sign up for upcoming events. I will now hand proceedings over to Dr. Tobias Karakach, who's our 2021 Early Career Investigator winner from Dalhousie University, to begin the panel. Thank you.
Thank you very much, Sarah. As Sarah has mentioned, today we're going to hear from world-renowned experts in precision medicine, utilizing big data generated by multi-omics technologies to guide the diagnostic therapeutic trajectories taken in medicine. To begin with, it's my pleasure to introduce Dr. David Malkin, who is a professor of pediatrics and medical biophysics in the Faculty of Medicine, University of Toronto. He holds the CIBC Children's Foundation Chair in Child Health Research, is a staff oncologist in the Division of Hematology and Oncology, director of the Cancer Genetics Program, and senior scientist in the Genetics and Genome Biology Program at the Hospital for Sick Children in Toronto. Dr. Malkin co-leads the Sick Kids Precision Child Health Initiative. He co-directs the Sick Kids Cancer Sequencing Program, which integrates next-generation sequencing into clinical care of children with cancer, and director of the Pan-Canadian Precision Oncology for Young People Initiative, which is establishing a pipeline to incorporate next-generation sequencing into novel clinical trials for children and young adults with hard-to-cure cancers across Canada. Dr. Malkin's research focuses on the genetic mechanisms of childhood cancer susceptibility. Recently, his work has addressed the application of genomics to early cancer detection and treatment approaches for children and adults with at genetic high risk for cancer. Dr. Malkin's research is funded by a variety of agencies, and he has published over 300 peer-reviewed manuscripts. He has received numerous awards nationally and internationally, recognizing his clinical research and mentorship work, most recently including the Queen's Platinum Jubilee Community Award, and being elected a Fellow of the Royal Society of Canada and the American Association of Cancer Research. Dr. Malkin, welcome.
So thanks very much, Tobias. It's really delightful and a wonderful opportunity. I think I'm looking forward to this session and having a great discussion and questions about this really expanding, burgeoning, and exciting field of precision medicine. And I will start with a sort of 15-minute overview of pediatric precision oncology, and as Tobias alluded to, touching on two of the programs that I'm involved with in Canada. One is Profile, the national program in precision oncology, and the other more locally at the Hospital for Sick Children, which I co-lead with colleagues Dr. Adam Schlein and Anita Villani, which is at the institution itself. So just to put this in perspective, childhood cancer is the most common cause of disease-related death beyond the newborn period. And in Canada, a country of about 40 million now, there's 4,300 approximately newly diagnosed children and young adults in the country each year. And of those, about a third of them will relapse from their disease or have refractory or metastatic disease. And this figure at the bottom here shows you adaptation of actually a slide, not a publication, from Stefan Pfister in Heidelberg, showing that at the time of relapse, almost any cancer that one considers, whether they be leukemias, solid tumors, or brain tumors, the overall outcome is grim, with less than 15% of children typically surviving. But cancer in childhood is really, at a genomic level, unique compared to adults. And so this paper, also is from the Pfister group from a few years back, using whole genome sequencing, demonstrates on the right-hand side of the panel that the number of mutations that occur in adult cancer, single nucleotide variants, is actually really typically very high, and sometimes in the hypermutation rate, sitting at sort of 100 or 10 or greater bases per megabase. Whereas in pediatric cancers, the mutation rate is relatively low. And in fact, many pediatric cancers are driven by fusion genes rather than point mutations in the genes themselves. There are some exceptions, and we'll touch on this a little bit in a few minutes, looking at this high-grade glioma cohort here, where pediatric high-grade gliomas, some of them have very high mutation rates. So one of the fundamental differences here is pediatric cancer and adult cancer are very, very different beasts. And this then leads to our need to think of different ways that we analyze them and generate data that is valuable in terms of clinical input. So in the KICS program, the so-called KICS Cancer Sequencing Program, the little logo at the upper left-hand corner of your screen, these are the enrollment criteria. These criteria are actually fairly consistent with current similar programs around the world, whether they be national or institutional in scope. Almost all of them enroll children with so-called hard-to-cure cancers, as I've touched on before. We also include children who may have a very rare tumor, for which the genomic landscape is unclear, and for which there may be very few or very inconsistent treatment approaches. And then as well, there's sometimes a very specific question about a patient sample that or tumor that we'll address in a second. Now, at the Hospital for Sick Children, this is the SickKids program, we've also included 3 other entry points. This is the primary one at the top. We actually enroll patients for assessment of cancer susceptibility. These would be patients who have a cancer associated with a particular susceptibility syndrome, or those who might have a strong family history of cancer that we are looking for an underlying alteration. We've also do RNA sequencing on all our leukemia patients now. And an entry point that just opened in the last couple of weeks, actually was approved, is the utilization of circulating tumor DNA for all cancer patients. And now all active cancer patients who are seen in clinic have blood drawn, plasma extracted on not every visit, but usually about every 3 months, to monitor their treatment, as well as to explore what alterations occur in circulating tumor DNA. We're heading towards the point, which I'll touch on in the very last slide, of implementing this comprehensive sequencing program for all children with cancer, not only at our institution, but right across the country. And now, in the last 5 or so years since the KICS program was initiated, we've had over 800 enrollments from 40 centers. And what I'd like to just point out here is not all these centers are Canadian. In fact, about 20% of the enrollments into the program are from international centers, including places like Memorial Sloan Kettering, Philadelphia, the Weizmann Institute, or Children's Hospital in Israel, and elsewhere around the world. So it really has expanded, if you will, the scope of patients coming through. It's Great Ormond Street in England, so on and so forth. Now, Profile, which is the national program, has a very similar scope, the main thing being it's national. And so the aim of the objective is to transform the care of childhood, adolescent, young adult patients across Canada by using next generation molecular tools, model systems, identifying disease and patient-specific biomarkers. A very similar objective to what we are doing at the local level. And in fact, when you look at the country, for those who aren't in Canada, it's a very large country geographically. There are 16 pediatric centers, which are highlighted here in the map itself, across the country, ranging from the West Coast right through the Atlantic provinces over on the right-hand side. There are 3 major sequencing centers in Vancouver, Toronto, and Montreal, and patients are enrolled in their local institutions, and then the samples are sent to one of the 3 based on geographic proximity. But in addition to that, because many variants that are discovered are unclear whether they're pathogenic or not, we actually model these variants in a variety of systems, both cell systems as well as zebrafish and mouse models. And these labs that do that are actually not necessarily in the 3 main centers, and so samples are distributed throughout the country to different research areas of expertise. And then in Calgary, out here in the province of Alberta, is where all of the data is centralized and the data repository is maintained. In addition, here are a list of a number of adult centers and cancer centers where adult patients are being seen, and adult patients, particularly in young adults, by our definition, that's up to the age of 39 years, are also referred into the program. And since it started about 6 years ago now, and with a slight dip during the pandemic, we've enrolled actually over 1,500 the typo patients from all these 16 centers. And the support from this comes from a variety of institutions and agencies throughout the country. So how do we do this? Well, in the KICS program, and to a certain degree nationally, Dr. Schlein, Adam, has developed about an 890 gene focused deep sequencing panel covering 890 cancer genes culled from various repositories, particularly relevant to pediatrics, but not necessarily. And then, of course, we do whole genome and, as I mentioned, transcriptome sequencing for all patients. The panel itself, here is 889 genes for the aficionados among you. It covers about 2.5 million bases. It's very deep coverage, and includes both well-known cancer genes as well as those that are perhaps more putative in nature. Now, getting back to the point about why we're doing it this way, well, so if we take the example of rhabdomyosarcoma is a common pediatric soft tissue tumor. It's the second most common soft tissue tumor in children. And this is a paper from a decade ago, but it's highly informative in this piece. So on the left-hand side of the screen, we see that there are patients with rhabdomyosarcoma who carry a driver mutation here. It's a fusion gene that drives this particular subset of rhabdomyosarcoma called alveolar rhabdomyosarcoma, fusion positive. And you can see there are very, very few other single nucleotide variants in other genes. There's a very quiet genome, if you will. Whereas this group of patients with a rhabdomyosarcoma that is not driven by the fusion, it's almost like every single tumor has a different genotype. And this tells us then that it's really very, very difficult in the pediatric space to come up with a small panel that will somehow or other identify the driver mutations or even passenger mutations in these tumors, because they're highly, highly heterogeneous from patient to patient to patient. Why is this important? Well, if we just look at a couple of genes, this is a Kaplan-Meier curve, survival curve of patients carrying mutations in the TP53 gene, and in here, what you can see is patients who carry a mutant P53 in their tumor have a poorer outcome than those with normal P53. And actually, when you start dividing it up among the sort of clinical risk, and so you do a multivariate analysis of clinical risk with the mutation, you can see that there are certain patients with intermediate risk, so-called, and P53 mutation, where basically the survival is 0. at least at about 8 years. So the identification of variants in this one gene alone in this particular disease makes a huge difference in interpreting the outcome, and that can also guide us in terms of selecting therapy. So the last few slides and few minutes then that I want to touch on are how do we use genomics at various time points in children with cancer. And so this slide, which I borrowed from Adam, really highlights the 5 major areas, and I'll give you examples of each of them and how they inform our approach to treatment and management. So the first, this was a study that was published early last year from our group, and Anita Balani led it with Adam and I and many others. In this, we basically sequenced the first 300 patients we sequenced in the KICS program, and found that about over half of them have at least one actionable finding on next-gen sequencing. And you have to realize all of these patients basically were out of treatment options. They had all had treatment fail them at least multiple times. So this is data that is consistent with other similar sequencing programs around the world, and encouraging in terms of next steps. What have we found? Well, in terms of predisposition, when you look at the textbooks of pediatric oncology, you sort of 5 to 10% maybe of children with cancer were thought to have an underlying genetic etiology. But we now know that to be a severe underestimate. In our hands, we're up somewhere around 20%. In this study, we were sort of around 17%, have germline findings across a multitude of genes. And what's interesting and important is that a number of these alterations are actually in the germline are potential targets for therapy. And so in the last slide, I'll touch on that for a second as why that does change the game for us. And here we see that about 15% had pathogenic or likely pathogenic mutations that were not previously known. A few had already been found. They might have been sequenced elsewhere. All of these patients were referred to genetic counseling, which of course leads to cascade testing for family members. An example of some of the curious findings, we've actually found a BRCA signature associated with DNA damage repair in a significant portion of these children. And while it's not entirely clear whether germline alterations in the BRCA gene are important or even directly related to the cancer they develop, and that's work that many people are exploring now, it does indicate that cascade testing to other family members is critically important, as especially older women, or at least their moms and aunts and so on and so forth. In terms of diagnosis, some very intriguing findings were found. So cytogenetic analysis is concordant with finding diagnostic tests. And so we were using a variety of tools. And this is an example of an alteration in a 10-month-old with a pelvic sarcoma, previously not otherwise specified, treated with conventional therapy, and was progressing on disease. And then when the KICS results came back, it turned out that they had an alteration in this particular gene called BCOR, which completely changed the diagnosis. And for those who are clinically inclined here, this is an MRI scan of this huge tumor in the pelvis, which was growing on therapy. Diagnosis was changed, therapy was altered, and after about 3 cycles of therapy, it had completely disappeared. And this child is now about 4.5 years since then, and is in complete remission and doing otherwise well. There's an example of a much larger study which was published last year, again by Adam's group, which led to a new diagnostic classification of childhood cancer using multi-scale transcriptomics, which is a critical way to go. And it touches on what I said earlier, is that single mutations in single genes are really not as informative as we think transcriptome is in the context of pediatric cancer. And then the next area is therapeutics, as I've told you. So this is an example of all the actionable mutations that were identified in a variety of the patients that we see, with the various genes along the right-hand side of the screen and the different samples along the top. And you can see sort of a scattering that everybody has, every individual has a mutation or an alteration in one gene or another, which could be targetable with drugs, but there's no two alike there. Everybody is different here. Every patient's different, which just goes to tell us how important looking at the entire genome is critical for for understanding the disease and also identifying potential therapeutic targets. This is one interesting finding that we've had at our institution, a great deal of interest in tumor mutational burden, which has always been sort of thought in the sense of melanoma, colon cancer, but Uri Tabori and Adam in our institution have found that about 9% of patients with childhood cancer have a high mutational burden, and these are exquisitely sensitive to immune checkpoint inhibitors, immunotherapy, and this has led to a number of clinical trials using immune checkpoint inhibitors to advance therapy in diseases as difficult to treat as glioblastoma multiforme, the very highly anaplastic brain tumors. And then the last section, as I alluded to, is the use of circulating tumor DNA to try to get a better handle as can we follow the evolution, the changes that occur in cancer cells as they progress, as they respond to therapy, as they relapse. Do they acquire new mutations? Do they drop out old mutations? And while many programs use sort of a single approach, what in collaboration with Dr. Trevor Pugh at Princess Margaret Cancer Center, he's developed a multi-omic approach, which we're now using panel sequencing, low-pass whole genome sequencing, methylation, T-cell rearrangement, and RNA-seq to really come up with a sort of smorgasbord, if you will, of possibilities at which variants can be found, and then use these to monitor patients over time and adjust the clinical management accordingly. So with that, leaving hopefully lots of time for discussion, I'd like to thank the entire SickKids team, both from the clinical perspective as well as our bioinformaticians and analysts, and two incredibly important people, our genetic counselors. And then as well, the national team, and needless to say, there are an awful lot of people involved here, and don't expect you to read them all, but this is how we've put it all together, and look forward to hearing any questions and comments that people have. So that's it. Thank you very much.
Thank you very much, David, for the interesting and very informative presentation. Our next panelist is Dr. Peter Campbell. He completed specialist training in hematology in New Zealand and Australia in 2002, and then went on to do his PhD at the University of Cambridge in the molecular pathogenesis of myeloproliferative disorders. In 2007, he took up a postdoctoral fellowship at the Sanger Institute in the UK, where he currently is a scientist. He's the head of cancer, aging, and somatic mutation, and a senior group leader, as well as a joint head of the Cancer Genome Project. He is also a practicing hematologist at Edinburgh Hospital in Cambridge. Peter and his team have developed a disease prediction tool that can give personalized predictions of future disease outcomes for patients with certain types of blood cancers. And his research focuses on the use of next-generation sequencing technologies for annotating cancer genomes and unraveling the genetic changes our cells acquire as we go through life, and how these mutations are related to aging, cancer, and other disease processes. Dr. Campbell made major contributions to the field of cancer genomics and how cells evolve across a person's lifetime, known as somatic evolution. His role in helping to lead the work of the International Cancer Genome Consortium has enabled the global research community to build a coherent understanding of the landscape of mutation in cancer genomes at the highest possible resolution. In 2019, he received a prestigious fellowship from the Academy of Medical Sciences. This award honors scientists whose research has advanced medical science, delivered cutting-edge research discoveries, and produced benefits for patients and wider society. Just last year, he has been awarded the Darwin Medal by the Royal Society. This prestigious medal is awarded to researchers who have made a significant discovery in their field through creativity, innovation, and intellectual rigor. Please join me in welcoming Dr. Peter Campbell.
Thank you very much, Elena. I'm just going to share my screen. Hopefully you can see that. So I'm going to present some data that we've been working on recently, looking at somatic mutations in normal tissues. You've seen the slide already. Basically, it kind of summarizes the journey that a cell goes through from the zygote through to the chemotherapy-resistant cancer, and during that kind of progression, you can trace the cell divisions from that cancer all the way back through the lineages to the fertilized egg. And during that time, it's been exposed to a number of mutational processes that introduce mutations into the genome. A few of those mutations, as we know, are responsible for the biology of the cancer. These we call driver mutations. And the typical cancer genome will have something like 10 to 20 driver mutations. But there'll be many, many more, thousands of them, typically, although as you just heard in pediatric cancers, often hundreds, of other somatic mutations that we call passenger mutations that are probably not related to the biology of the tumor. And we've been through the advent of massively parallel sequencing, we've been able to characterize these cancer genomes in considerable detail, as Elena outlined. What we haven't been able to do, and what I'm going to talk about today previously, is characterize what's happening in normal tissues during those very early stages of cancer evolution. We've had limited insights into what this kind of pattern of mutations in normal tissues looks like. Through a series of kind of technological developments, we've now been able to do this across a large number of tissues now. And I'm going to show you some of the kind of common themes that emerge in terms of the patterns of somatic mutations in normal tissues as we age. And the first, and in some ways, one of the more dramatic findings is that the mutation burden pretty much increases linearly with age in all tissues that we've looked at. So the rate varies from organ to organ, as you can see here. You're looking at something like 15 to 20 mutations per cell per year in a blood cell, more like 40 to 50 mutations per cell per year in the colon, and then lots of other organs show sort of intermediate levels as shown here. And that linear increase in mutation burden with age is present even in differentiated cells like lymphocytes. It's present even in post-mitotic cells like neurons. So it really is a universal process that's happening in all of our cells all of the time. Those mutations, they begin right at the beginning of life. So this is data from an 8-week fetus that we published, where we took individual blood cells and then grew them into colonies and sequenced them. And then we can kind of look at the patterns of sharing of mutations and trace the lineage relationships of those cells back in time. And so this is the sort of phylogenetic tree that we observe from this 8-week fetus. Each of the tips of the tree here represents the genome of one of those blood cells that we sequenced from the fetus. And you can see by tracing back the pattern of sharing of mutations. And what's really interesting and instructive is to look at the top of the tree here. So you can see that there are kind of branch points early on in this phylogenetic tree that essentially divide the population of cells into 2 and then into 4, etc. And the mutations that kind of make up these branches, we actually found in all tissues of that fetus that we sequenced, including, remarkably, the placenta. So what that tells you is that the cell divisions that underpin these branch points in the phylogenetic tree predated the split between the embryo proper and the placenta. And therefore, they must have been incredibly early. And in fact, we think that these mutations that split this tree represent the mutations acquired in the first cell division of the zygote. So right from the beginning of when you're a fertilized egg, you're already beginning to accumulate somatic mutations. One of the other most striking things to emerge from this data is that our normal tissues are riddled with driver mutations. So the mutations that we might see in a cancer genome. So this is data from my colleague Inigo Martin-Carrera, and in which he sequenced patches of normal esophageal epithelium from 9 individuals across the age range that you see here. And basically what you see here is a mock-up of what 1 square centimeter of esophageal epithelium looks like. And the circles in these mock-ups represent individual clones. And when the circles are empty or white, they don't have any driver mutations in them. When they have a color in them, that's a clone with a driver mutation in one of the genes shown at the bottom here. And you can see a number of interesting patterns in this data across the age range. The first thing is that with time, with age, the number of clones increases. So there are more circles here compared to here, and those clones are much bigger. So the circles are bigger than they are early in life. And you can also see that many more of these clones have driver mutations in later life. And some of these clones actually have 3 or 4 driver mutations later in life. And so what that tells us is that the kind of fraction of cells that has driver mutations increases steadily with age. It actually increases more than linearly. It increases quite substantially, so that by the time you're in your 50s, 60s, and 70s, the majority of your cells, 70, 80, 90% of your esophageal epithelial cells will have driver mutations. And these are the sort of driver mutations that we would definitely expect to see in an esophageal squamous carcinoma. So these are probably cells that have taken their first step on the path towards malignancy. And it's the same in other organ systems. So we have data from blood here, where we can see this kind of dramatic change in the clonal architecture of blood after the age of 70. So in people under the age of 70, what you see is that the phylogenetic tree essentially looks like a comb. You get lots of individual stem cells that are contributing to blood, probably 100 to 200,000 at any given moment in time, and they're all broadly contributing equally. But after the age of 70, you see this universal pattern where you have a small number of independent clones that are contributing large numbers of blood cells to the mature blood population. And it's around about 30 to 60% of blood production in people over the age of 70 can be accounted for by as few as 10 to 20 independent clones. So there's this kind of oligoclonal expansion. Many of these clones have driver mutations that we would typically see in a leukemia or other blood cancer. But many of these expanded clones don't have any recognized driver mutations, and we believe that there are many more driver mutations in blood that have yet to be discovered. And these driver mutations are not restricted to normal aging. We also see them in the context of diseases other than cancer. And so this is data from patients with metabolic liver disease, where we take patients who, this is a sort of obesity-associated metabolic liver disease, where you get fat accumulation in the liver. And here we see this pattern that, again, we have lots of independent clones with driver mutations. So here's a patient at the top who has 5 independent mutations in a gene called FOXO1. FOXO1 encodes the transcription factor that's the master regulator of insulin signaling. And essentially these mutations render FOXO1 resistant to insulin signaling. So the hepatocytes become resistant to insulin. And you can see that this patient has 5 independent mutations in different clones, all in the same gene, in fact, the same hotspot. This is another gene, GPAM, in which we have a patient here with 7 different mutations, all in GPAM. And these mutations are all predicted to be loss-of-function mutations. GPAM is the master regulator of converting free fatty acids to storage triglycerides. And so it's a key member of the lipid handling pathway in hepatocytes. And here's another patient with 3 different mutations in a gene called SIDE-B. SIDE-B is involved in lipid droplet fusion in hepatocytes, and these mutations are predicted to be loss of function. What's fascinating about this data in chronic liver disease is that these mutations, we have this pattern of within patient convergence. In other words, within a patient, we see the same gene being hit again and again and again, 7 mutations in GPAM within the one patient, but between patient divergence. So in different patients, you see different genes being hit. And so what we think is going on here is that the disease process that these patients are exposed to, the fat accumulation in the liver, is essentially killing a whole bunch of the hepatocytes within the liver. So you might start as a nice, healthy, very polyclonal liver with lots of different mutations in all of these different genes. Along comes your disease process, and it kills all of the genes except those that have some protective mutation. And those protective mutations in this case might be the pink mutations. And so the cells that survive are predominantly made up of pink clones. And then what happens over time is that these are the cells that recover and regenerate to repopulate the liver. And then when we come along and sequence it, we see this pattern that we see lots of independent clones with pink mutations. In a second patient, the disease process might be subtly different, so that in this second patient, it's not the pink mutations that protect the cells against the disease process. It's instead the blue mutations that protect the cells. And so when we sequence them, we only see the clones that have the blue mutations. And what that tells us is that the selective pressures on these cells are subtly different in the two cases. So for example, this patient, the one with the pink mutations are protective, might get to their liver disease through excessive alcohol intake, and that leads to de novo lipogenesis. So any pathway that protects cells against or switches de novo lipogenesis off are going to be protected against that form of lipid buildup. But if you get to your lipid accumulation in your liver through eating your fat, like eating lots of pizzas and other fatty foods, then de novo lipogenesis isn't really relevant to you, and instead you're going to the genes that mutations that protect you against insulin signaling would be more likely to be selected for, and so we see a different pattern of mutations in those patients, and that's what we believe is going on. And then this is a third instance where we see these driver mutations accumulating with age. This is in endometrium, and here's a 34-year-old woman where we've sequenced individual endometrial glands, and every single gland that we sequenced had at least one driver mutation. Some of them had two driver mutations. It's really quite a striking pattern. All of these genes would be regularly seen as mutated in endometrial cancers, and they're already present by the age of 34 in every single gland in this patient. Why this patient has so many of them? We sequenced other 30, 40-year-old women, the endometrium from other 30, 40-year-old women, didn't see nearly the same penetrance of mutations as we see here. Why this woman is different, we have no idea. So I'm just going to kind of summarize some of this data and draw out some of the general themes. The first is that this background mutation rate that is present in all of our cells leads to spectacular genetic diversity within an organ. So for example, the number of T cell clonotypes in one person is about 10 billion, and the average mutation burden per T cell at the age of 60 is 2,000. What that means is that every single base in the genome is mutated about 6,000 times on average across T cells, and that 10,000 mutations are acquired every second in your T cells. And it means that there's more genetic diversity in one subcompartment, i.e. T cells of one organ, the lymphoid system, of one individual, than there is inherited variation across the whole human population. So there's this spectacular genetic diversity within our organs. And what then acts upon that background genetic variation is this pattern of evolution selection for particular mutations within tissues. And it turns out that this selective pressure is much more critical to defining the clonal structure of tissues than the actual background mutation rate. And those selection pressures can take many different forms. We see organ-to-organ differences in what driver mutations. We see aging, as I've shown you, clearly has an effect. Diseases like the chronic liver disease has effect. We see changes in people who smoke compared to those who don't. Medications, germline variation can also have effects in shaping the clonal architecture of tissues. Much of that evolution never leads to cancer. Those mutations that I showed you in the liver, the FOXO1 mutations, the GPAM, the SIDE-B mutations, they're frequently mutated in chronic liver disease. We barely ever see them in hepatocellular carcinomas. So not all of this somatic evolution has to finish at cancer. And indeed, not all of it is bad news. So this is a phylogenetic tree from a 2-year-old boy who has a ribosomopathy caused by DNA JC21 germline mutation. And what we see when we look at his blood cells is this recurrent mutations in genes that protect the cells against the ribosome defect. And in fact, this boy would not be alive if it weren't for these blood cells that had found some way of evading the germline defect. He would not be able to make blood except for these cells that have essentially rescued his phenotype through the process of somatic evolution. So that's where I'm going to finish. I just want to say that I think that understanding this, measuring the kind of clonal architecture of tissues in this way gives us really fascinating insights into all of the forces that are shaping cancer evolution. And we believe that this might be a way to think about risk prediction for who's going to go on and get cancer, and also to measure how effective potential preventive therapies are in reshaping that clonal architecture and maybe pushing the clonal architecture away from cancer-prone structures towards more benign patterns. Thank you very much.
Thank you, Peter, for your presentation. It is my pleasure now to introduce you our next speaker, which is Dr. Kym Boycott. Dr. Kym is a professor of pediatric at University of Ottawa. And also, well, she is also a clinical genetics at the Children's Hospital of Eastern Ontario and senior scientist at the Research Institute of this institution. Dr. Boycott's research program is in rare disease, which aim to bridge clinical medicine to basic research. And he is also focusing on understanding the molecular pathogenesis of this disorder to improve patient care and family well-being. She leads the well-known National Care for Rare Cancer Consortium, integrating genomics and omic technology to improve our understanding of rare disease, with a particular focus of solving the unsolved and most difficult rare diseases. To leverage these discoveries, she co-leads the Canadian Rare Disease Models and Mechanism Networks, which has been established to catalyze a connection between newly discovered rare disease genes and basic scientists who can rapidly study them in a model system. Globally, she moves the rare disease agenda forward through a role in the International Rare Disease Research Consortium, the Global Alliance for Genomics and Health, and the Global Commission to end the diagnosis odyssey for children. So it is our pleasure today to welcome you, Kym. Over to you.
Thank you, everyone. It is a pleasure to be here and lovely to follow David and Peter. I think we're each taking very different approaches and covering a broad range of biology that should be particularly interesting to our group of listeners. So we're going to switch it up a little. And I'm going to focus on rare disease precision health. And the angle I'm going to come from, I think, in my 10 or 15 minutes of comments is really around the challenges with data coordination and data sharing with respect to precision health. And I think from it's really coming from a rare disease perspective because it's so important in rare disease, and I'll explain why that is. And I'll really shape my comments around our experience in the Care for Rare Canada national network and Canada's All for One Precision Health Partnership. So just starting with rare genetic disease for our audience, rare disease itself probably affects 3 million people worldwide. When we use the term rare disease, we're talking about all kinds of diseases, including rare cancers, which we've heard about from David, as well as autoinflammatory diseases, etc. When I talk about rare genetic diseases, I'll be talking about those that are really caused by dysregulation of a single gene. So a little bit different than what we heard about from Peter, where many driver mutations are happening. This affects 1 to 3% of the population. And these rare genetic diseases are quite well recognized. There are more than 6,000 caused by dysregulation in different ways of more than 4,000 genes. And it is important to recognize that there are thousands that remain unsolved in 2024. And what this presents as a challenge to patients and families is what we refer to as a diagnostic odyssey. And it's depicted here, which really sort of describes something that can take, on average 5 to 7 years, as long as 20 to 40 years for patients presenting for the initially, or families presenting initially, all the way to an accurate molecular diagnosis. So in order to facilitate an accurate diagnosis, this is the key to precision health in this and really any area of medicine. And what we need to do is twofold. One is we have to facilitate access to the appropriate testing at the appropriate time in the diagnostic care pathway. But the second thing is we have to be able to understand the molecular pathogenesis of all the rare genetic diseases. And as I mentioned, that knowledge compendium is certainly far from complete at this time. And so we've had clinical genome-wide sequencing as a diagnostic test. This largely refers to massively parallel sequencing of all the coding genes in the genome at one time. And this can be achieved in a couple of ways, including exome or genome sequencing, because genome sequencing, quite honestly, at this time is really mostly analysis of the coding genome. This is the best test we've ever had in genetics and probably in many areas of medicine, with the diagnostic yield upwards of 37% depending on the patient population. Now, hundreds of thousands of rare disease patients have been analyzed using these technologies worldwide, and we have variable but improving access to this genetic testing across Canada. Now, as I said, a diagnosis depends on discovery. So you can see the little girl there in pink. She represents a diagnosis. And there are a number of reasons why we may not be able to identify a diagnosis. As I said, that diagnostic yield is about 37% or so. And I'm going to focus first on the purple girl hiding behind the tree. And that is that the disease gene association itself has not yet been discovered. And how do we approach this particular challenge? And this really comes down to national infrastructure for data sharing. And the reason that this is so important in rare genetic disease, is that we are possibly looking at one or a handful of families with a particular rare genetic disease in Canada. And so without sharing across our country and with other countries, we really won't make any advances. And I think some of the really nice work we've done over the last 10 years in discovering these new disease gene associations, it's all been about international data sharing. And so the platform we developed is called Genomics 4RD. It is a research infrastructure, and it allows clinical data and omic data, all kinds of omic data, to be brought together and collated from across Canada and shared in an open, controlled, or restricted fashion, depending on what that actual data is. And it allows us through the right side to go out internationally for matchmaking and discovery. And, I'm also want to highlight at this point that, this data infrastructure took us 5 years to build and populate. And I have an image of it here, not that I want you to look at the complexity of all these pieces, but just from a high level to appreciate this kind of infrastructure takes a long time to put together. And it is a very thoughtful process. And now we have 315 data depositors that are working in this area, with almost 8,000 omic data sets that are being shared. But investing in this kind of infrastructure, and more importantly, maintaining it after the grant is finished, is of ultra importance if we're going to make any significant advances in precision medicine. Now, what this kind of infrastructure lets us do is matchmake. And so of this 65 to 70% of patients that we're not solving, why aren't we solving them with the genome-wide sequencing? And this infrastructure allows us to take on many types of matchmaking. And matchmaking is what we refer to making an association between a gene and a set of clinical phenotypes and variants in a gene for the first time. And so you can see there's two-sided, one-sided, and zero-sided matchmaking. I'll focus on two-sided, but there really is multiple ways to do this. Just to highlight, zero-sided matchmaking, for example, is really AI type driven or machine learning type driven processes that allow you to take huge data sets that are clinical and omic and put them together and try to make associations between variants that you're seeing and a disease state. One-sided matchmaking refers to people coming into your data set and looking through it to see if they can help facilitate a hypothesis that they have. And two-sided matchmaking is what we've lived on in the rare genetic disease space to make discoveries for the last 8 years. And I'll show you an example of that. So we recently, 2002 or 2022, we wrote a review on Matchmaker Exchange. So now it's been 8 or 9 years since we launched this initiative to connect databases that contain unsolved rare genetic disease patients from across the world. We currently connect 134,000 patients from 104 countries. And within this system, we have more than 15,000 genes tagged as someone being interested in them. And two-sided matchmaking is like a game of Go Fish. You're basically putting it out to the international community. Do you have any patients who have variants in gene X with clinical features Y? And so this is Justin's story, and he has been a patient at my institution since he was a baby. He's now 28 years old, and he was not diagnosed until he was 25. And he had candidate genes identified after exome sequencing, identified in the research setting, but there were 2. And so what we had to do then is put both of those genes out into the Matchmaker Exchange. And so we include phenotypic data and genotypic data, and we launched that through to see if we can connect with anyone in the world. So the short story of this is that initially we matched with a group of, researchers around the world, clinician investigators, because Justin had a de novo, so new change in him, variant in the gene USP7, and those are represented by the green dots. And so there were many individuals around the world, and they had features that overlapped with Justin's. But the issue was that Justin had MRI imaging features with white matter changes that none of those other green dot patients had. And so we know what part of him was explained by the USP7 variant, but the rest of him potentially was not. And so a year later, we matched with the yellow dots in TMEM 106B. And basically, they all had the exact same de novo change that Justin had, and they all had the same MRI imaging. So Justin's clinical presentation is because he has 2 de novo novel gene disease associations that were discovered sequentially and could not have been done so without this sort of data sharing infrastructure. So over the last decade, the Care for Canada Consortium has identified this lovely set of genes that I'm showing here, and they're mapped to where those genes operate in terms of their biological processes. All of these discoveries were facilitated by international data sharing. Not a single one would have been a discovery that could only have been achieved in Canada. So moving now to the fact that we have this coding genomic data on all of these patients. What if that still doesn't give us the answer? And we have found that in 30 to 40% of patients, that is the case. Even if you look through all that coding data, you're not finding a compelling explanation for their undiagnosed rare genetic disease. And so in this case, perhaps it's like this little guy in the box, the genetic etiology is undetectable to that technology or uninterpretable. And so this is where we talk about the next wave of omic technologies and how we're using them. We heard about transcriptome sequencing from David. So we've been using that as well as deep sequencing, epigenomics, metabolomics, lipidomics, and long-read genome sequencing to take all of our unsolved patients who are coming through the pipeline, and try to see if we can find an explanation. And so quickly, what we learned is that when exome sequencing came into the research setting earlier on 2012 was when we started to use it as part of our National Care for Rare project. It was pretty straightforward. You collect and consent, engaged families, clinically well characterized, you perform exome sequencing, you share data, you find stuff. Multi-omics is different. And I really like this image of the crisscrossing tracks. Multi-omics is going to require even more novel computer approaches on how to analyze that data, and even more data sharing and data platforms required to help us interpret that. And so what we did was take just almost 1,300 families who had exome sequencing, but reanalysis, and then went through a reanalysis pipeline. We then started applying short read RNA sequencing and long read genome sequencing to this group. And what we found was actually quite interesting. So when we reanalyzed the coding data for this group of patients, 15% got a diagnosis. And a lot of that was because of some gene discovery that had happened in the years since the first diagnosis. All of those that were undiagnosed at that point went through the additional omics. And of that, another almost 14% were diagnosed. That additional omics piece requires a lot more intensive data analysis and data sharing and time than the first 15%. And so the gain is equal, but moderate. So when we think about now some of this genomic sequencing in the rare disease space, it's being used in clinical care. And so how do we leverage that clinical data and that genomic data for precision health? Because what's happening now is that all that diagnostic testing is being siloed amongst different institutions. And so how do we prevent that, especially in a country like Canada, where there are lots of institutions who are data custodians of this data, and we really have no experience or mechanism by which to share that data. And so this is where the All-for-One Precision Health Partnership comes in. And what it's trying to do is build regional genomics capacity across our country, promote equitable and ethical uptake of these precision health tools, and address barriers to data sharing. And so just quickly, so we can wrap up and have some time for questions, this is a Genome Canada funded initiative, and it is funding these demonstration projects across the country. And the idea then is that we're going to sort out a way to facilitate precision health in Canada using clinically generated clinical data and genomic data. And so this is an example of Genome-Wide Sequencing Ontario. This is the one I lead with Martin Somerville from SickKids. And we began this in April of 2021. We've already sequenced almost 7,000 individuals. And the concern there is that the data could potentially become siloed at my institution and at SickKids, not even sharing with each other. And so despite the amazing successes with the 30% diagnostic yield, et cetera, and a great turnaround time, we needed to have something else move forward. And so what we developed was a health data ecosystem, and we engaged with over 100 different stakeholders to talk about how we might do this in our country. And we focused on a clinical arm and a research arm. The clinical arm is what we refer to a national network of diagnostic labs. So these are trusted laboratories that are interpreting this testing, and can they share with each other. And a research arm, which would function as a national registry to recontact patients to share their data for secondary use. And this would allow us to provide equitable access to precision health research for Canadians, no matter where you live. And so we don't need to look at the design at all, except to say that this took us 18 months to develop. We are now able to share data with SickKids in Toronto, and we're bringing on BC and Halifax shortly, so that'll be great. It will be the first in Canada way of sharing this sort of clinical precision health data to help interpretation for patients no matter where they live. And the research arm is being developed in collaboration with CIHR, which is our Canadian Institutes for Health Research, as well as Genome Canada, to talk about how we'll be able to leverage that data for secondary use. All this image really is to say that the complexities of how to set this up really is going to be regionally country specific, because we all have a different set of challenges, though there are things that we can definitely learn from one another. And finally, I want to show 2 slides on rare disease precision health in action. And so this is a little girl that was seen at my institution who was identified to have a de novo mutation in GRIN2A. And over on the right-hand side, it's showing an in vitro assay that we performed with collaborators to look at her specific variant and then use that in vitro assay to assess which anti-epileptic medication might be best for her. And the reason this was so important for her is that she had over 200 seizures per day. And so she was put back on, she was switched over to memantine, which is a very old drug, which is not used for epilepsy very often. And she had a reduction in seizures of almost 100%. And that reduction was maintained since this time in 2014, which has been quite remarkable. And the other example I want to show you is this is a novel gene discovery that we identified a few years ago. And this is Ernie. And you can see he's got a very small cerebellum here that is atrophying over time. He has this finding, which we found after we identified the gene, which is a manganese transporter, that he has very, very low manganese in his blood and he's wasting it in his urine. And so we are now running trials and interventions in Canada and with our international partners, supplementing these kids with manganese. And some of them now are being detected right at birth because this condition is present in one of our founder populations here. And as a result, we're being able to treat these kids right away and hopefully prevent a lot of damage in the long run. And so finally, I just want to thank this large group of collaborators that we've been working with. The things I'm telling you about are the output of hundreds of scientists and students and clinicians and patients and families from across the country through many of our big, large-scale projects that you can see indicated across the top. And all of our funders are down the left and the right-hand side. So thank you.
Thank you very much, Kym, for this interesting presentation and for all the work you are doing to help patients and their families. So we are now in the period of what you are waiting for, the second part of our meeting today, which is the discussion. So thank you, David, Kym, and Peter for your very interesting presentation, for giving us an overview of what we can do in precision health. And for the audience, if you have questions, please use the Q&A chat to address your question. I will be happy to share with our speaker. I will claim the moderator's privilege to ask the first question, if I may, and it will be a general one. Even if you took a different perspective, one common aspect I can see from this is collaboration, right? So I wonder if you can elaborate a little bit more on how collaboration is important in this field, particularly if you have an advice for the early career researchers entering in this field. So I will start with you, Kym, please.
Sure, mine's the easy one. So I think, you know, in the rare disease space, as I was saying, that if you don't collaborate, you can't be successful. So it's really ingrained in our culture. And I think it's driven a lot by the fact that the clinical teams really want to find diagnoses for their families and help them sort of guide their management based on that information. So it's honestly, it's quite natural, and we've been doing it for more than 10 years. And honestly, I think if we looked once in our Care for Air Canada consortium that published with more than 1,000 different authors in I don't know, 40 countries or something like that. With respect to early career investigators, I think that's a great question. And I think, you know, how we like to approach that is a lot of our large-scale projects that we lead, it is very important to support and have a very specific place for the early career investigators. And now at my current age, I find that even more important. I used to be an early career investigator, and now I'm definitely the senior one. And because we have to have succession planning. And so we're planning, we've got all this great infrastructure that we've built, these collaborations, but I'm not gonna be doing this, for the end of time. We have to build that next generation to be able to take it on and continue to support the work that we've done here.
Okay, thank you. David?
Yeah, I think I would agree with everything that Kym said. In the pediatric cancer space, pediatric cancer is essentially a rare disease, although it's sometimes not defined as such because it's cancer. But if you subgroup them, as I sort of was alluding to in the way that the genome analysis is shown, is that these are all subdivisions of subdivisions of subdivisions, and they all have to be dealt with differently. So similarly, the necessity to do large-scale collaborative work in genomics and also the clinical trials that result from those is the only way for advance going forward. I think childhood cancer has probably been it's always touted as one of the best examples of where collaborative work over the last half century has led to some of the advances that we see that now other disease entities are kind of sort of following along. And I don't think I have much to add to Kym's comment about young investigators. Absolutely. I think our team, if we looked at that, those two slides, you couldn't see all the names, but I think there's a fairly equal mix between those who would be considered senior and those who are mid-career and those who are coming out of their training and building their careers. And just one small, what's it called? Fun fact is Adam did his postdoc with Peter. So there you go. Which may explain partly why one of our slides was identical. I think actually that was yours, Peter. It says Adam sort of took it over and then.
Interesting. Peter, what is your take on that?
Yeah, no, I think in this world, the sort of complexity, as David was saying in his presentation, the kind of variation from patient to patient in the kind of patterns of genomic changes in cancers means that one needs massive sample sizes to be able to infer the consequences of any given driver mutation or any other genetic change. And to get those massive sample sizes really needs collaboration. No single center can do it. I'm not even sure that a single country can do it. Maybe some of the bigger ones can, but we really need thousands of patients with a given tumor type to be able to make accurate inferences on the prognostic implications of the genome sequencing.
Thank you. We have a quick question on the Q&A. Which is, are all genetic tests publicly funded in Canada? I'm wondering, patient or family cannot afford this test and can refuse to get a precision test. I think this question is for you, Kym and David.
Yeah, I think so for both of us because it's a little bit different. So yes, genetic testing is publicly funded in Canada, so it's accessible to anyone where that test is approved to be funded in that particular region of Canada, which it's pretty standard across the country.
So the modification there is genetic testing for germline is publicly funded. Genetic testing for somatic disease is not as yet. So in the cancer space, that's of course a big deal. And we and others across the country are working towards that. Interestingly enough, last year, the province of Quebec actually has now provincial funding for whole exome and RNA sequencing of cancer patients' tumors with paired germline going along with that. We're working towards that in Ontario and hopefully spread it around the country. In terms of clinical trials, it's a little different. I mean, clinical trials are by their very definition investigational, so they are all funded. So technically funding should not preclude anybody in Canada from participating in a clinical trial. There may be other reasons like distance and so on, but funding is actually not one of them.
Thank you for this clarification. Peter, maybe you can jump in and provide the perspective from UK, for example, regarding this question.
Regarding the sort of how
the
genome sequencing is entering the clinic, so we have kind of panel sequencing available for many tumor types within the NHS, so it's a publicly funded health system. I think genome sequencing is still considered sort of somewhere between kind of research and clinical practice, there are a few tumor types for which whole genome sequencing is available on the NHS through the auspices of the Genomics England.
Okay, I'm wondering, this is a question for you, Kym and David, how omics in rare disease can inform precision oncology medicine and vice versa. I know that there is some, common ground you can find around this type of two type of medical conditions. So I'm wondering if there is, how one can inform another.
Going to go first, David?
Yeah, sure. I think I think the message that I was trying to impart in mine is that the omics, however it's done, is starting to inform some clinical management decisions. So, for example, of the 56% that I said in the Kingston, and actually the profile study is very similar in other studies around the world. So 56% you can find an actionable target, meaning a new drug, it's a target for a drug, it's genetic susceptibility and various things. However, of those 56%, only a small fraction is there actually a drug available to target. So that's the challenge now, is we actually are understanding that there are a lot of biologic targets that we're identifying from genomics. Now we've got to figure out a way to identify and push all these drugs to them. The other thing is that it's a somewhat simplistic picture. Actually, I really like Kym's model of the one train track versus the multi-train tracks, because this doesn't really take into account things like polygenic risk score, or when you get variants of uncertain significance across 3 DNA damage repair. Well, each of them one may not in and itself explain the problem, but you kind of imagine, well, gee, all 3 of them are messed up, then something's going on here. So we really need a lot more understanding of what the functional consequences of all these alterations mean, to then dissect out what the most appropriate management strategy is. But there are definitely examples, and the one that I highlighted was the increased tumor mutational burden in disorders that immediately lead one to start immune checkpoint inhibitors, and those have made significant inroads in management. So yeah.
Thank you.
And I would say, I think in the rare disease space, that less than 5% of rare genetic diseases are treatable. That is a well-known number. And so the patients that are reaching the diagnostic point in their care pathway where they would get a analysis of their coding genome don't have the easy stuff we would have figured out beforehand, which often have the tailored treatments. And so I think that's an important thing for people to recognize, that by the time they get to needing such a large scale, broad look at their genome, it's often not going to be associated with some kind of therapy that wasn't obvious before, though there are exceptions, which I showed you in my remarks. And so what we're really trying to do then is tailor their management based on our understanding, and making them, helping them become visible in both the healthcare system and the community system in terms of access to resources.
Yeah, thank you. So now, Peter, a question for you. This is related to duplication of gene. This is a question from Elena. She wants to know if you think that the duplication of gene poses a challenge to precision medicine, since duplicated gene represent genetic redundancy, a potential mechanism for compensation that may well lead to low therapeutic efficacy, side effects, and also resistance to treatment. What do you think about this?
Yes, I think that's a very interesting question. In fact, I would say that, when one talks about gene duplication, it comes in many forms. And I think that, from a simple 10 of duplication that just duplicates an extra copy of the chromosome, to kind of massive amplifications of genes through more complex processes. And what we know about this landscape is that it's incredibly complex within cancers. So the patterns of structural variants that one sees across cancers is really very diverse, and there are many different mechanisms reshaping the cancer genome. In fact, when we look in the normal tissues, we see very little of the structural variation. So it's one of the major differences between normal tissues and cancer genomes is the presence of the structural variation in cancer genomes. What it means for precision medicine, I think, has barely been scratched. We have typically in the clinic focused on point mutations in known cancer genes as the kind of handle that we hang our precision oncology on. But it's missing a large chunk. We believe that probably in many tumor types, much of the variation, much of the biologically relevant genetic variation is structural, not point mutation driven. So for example, in the sarcomas and so on, many sarcomas are driven more by structural variation. Ovarian cancer is driven more by structural variation than point mutations. And we really need to understand how to describe this landscape of structural variation in order to work out what it means for a patient. What does it mean to have 10 copies of MYC? What does it mean to have a structural variation affecting CDKN2A and in various different tumor types, as well as point mutations in KRAS or whatever? And I don't think we really have the language for describing these patterns of structural variation or the algorithms for annotating a cancer genome for what's the relevant structural variation for a patient. And we certainly don't have the clinical kind of knowledge banks to work out what these these structural variants mean for prognostication, for treatment response, for drug resistance, as your question kind of gets at. And so part of the reason and the desire to move from targeted panel sequencing to whole genome sequencing in the clinic is so that we can begin to build that knowledge base of what structural variation means for patients.
Okay. Thank you. David and Kym, do you want to follow up on this?
Go ahead, David. You look like you're mid thought there.
No, you go ahead. I'm mulling.
I was mulling as well, so no specific comment from me.
Okay. A question from Janet. This is for Peter as well. I'm intrigued by the selection for somatic mutation that actually protect from disease. How prevalent do you think this is? And also, Kym and David, you are feel free to follow, of course.
All right, Peter, you're up first.
Me first? Okay.
Yeah.
All right. Well, I think it's a really interesting question. It's clear that the selective landscape that we see in these different settings is both quite plastic. So we see, for example, if you kind of give someone a therapy, you can if the therapy's reasonably active, you can see a complete rewiring of the driver mutation landscape in that tissue as a response to the pressures exerted by that therapy. So, and we know that, for example, if you stop exposure, so if you take lung epithelium from a smoker, you'll see lots of p53 mutations, Notch mutations, FAT1 mutations, typical things that you'd see in the squamous cell carcinoma of the lung. If you stop smoking, what you see is this emergence of cells that are essentially wild type, that they don't have these driver mutations, that the fraction of cells with driver mutations decreases after you stop smoking because the wild type cells are probably better adapted to the non-smoking environment, whereas the cells with driver mutations are better adapted to the smoking environment. So what this tells us is that the landscape of mutations that we're seeing in normal tissues from which the cancer emerges is very plastic and very fine-tuned to the particular environment that a patient is exposed to. And we know that these different driver mutations, as Janet was kind of hinting at in the question, have different kind of rates of transformation to cancer. So you can imagine that if you had an esophagus or a lung that's full of p53 mutant clones, that's much worse news than if you have an esophagus or lung that's full of either wild type cells or Notch mutant clones. And we are seeing this, and this is backed up by mouse models as well. So what we would really like to be able to do in the future is to identify kind of therapies or approaches or lifestyle changes that nudge the landscape of a normal tissue away from, you know, lots of nasty clones with p53 mutations, et cetera, and towards kind of patterns which are either more wild type or more benign driver mutations. And we think that that should be possible with even relatively simple nudges. And you probably don't need to wipe out all p53 mutant clones. You probably just need to nudge it, you know, drop them by 10 or 20%, and already you'll begin to see an effect on the patient's risk of cancer.
I might just add or elaborate very briefly as well as this whole concept of resilience genes, or at least resilience in patients who carry germline events. And, you know, we know the classic one, Delta 508 mutation in cystic fibrosis, and there's people who are living very long lives who have absolutely no symptoms of cystic fibrosis. And there's many examples of these things suggesting that are there modifier genes in the germline? Are there epigenetic events or other things that are modifying? And then getting to Peter's point of allowing for a different route to transformation of the cell, whether it's cancer or many of these other diseases. So it's really quite fascinating when we think of the variance and variability and penetrance of virtually all these genetic events is very dependent. Even a simple one like retinoblastoma, everybody for the longest time thought you carry a mutation RB1, you're going to get retinoblastoma, but it's not quite like that actually. And nobody knows what all the other events might be. It's really intriguing.
Yeah, and I think from a rare disease space, we probably don't really understand how much rescuing is going on in some of those conditions, but there's definitely some that we can see, and there's a handful of conditions where sort of rescuing is part of the mechanism of disease, or rescuing, you know, it's sort of turning it off, turning it on, changing something. So I agree that, you know, the cells are very plastic. A lot of stuff happens when the environment changes.
So, Peter, David, Kym, thank you very much for sharing your insight with us. We are running a little bit on behind the schedule, so I pass over to my colleague Kym for the closing remarks. Thank you very much for this.
Thank you very much for the wonderful discussion. And we heard today about pediatric cancers that are driven by fusions, not point mutations, from David Malkin, and that child cancers represent rare cancers that really present themselves in hundreds of mutated genes. And many of those are germline mutations, so they're really inherited, making them quite a bit different than adult mutation spectrum. We've heard from Peter Campbell the evolution of mutations starting from fetal stage onwards, and how the various disease states may actually change the patterns of these mutations through selective pressures, and the idea for future of precision medicine is really to start pushing the mutation patterns away from those that transform cells into malignant states. And finally, we heard from Kym Boycott, who really talked to us about rare genetic disease and the sort of data sharing platforms that are enabling discoveries and diagnoses of undiagnosed diseases, and the parallels that there are with cancer precision medicine. We talked about the need and to build access and equitable access to data across Canada, and between Canada and other countries, to build access across multiple centers, across multiple provinces, to really to push forwards the progress in precision medicine. We heard how important it is to collaborate, how huge sample sizes that are really necessary for making progress in this space are really enabled by these collaborations. We heard about the public funding that's going in to support the analysis of germline mutations, of somatic mutations. That's how it's expanding to benefit the patients. And the functionally annotating the genomes that came out of the discussion. That's important. Focusing on variation other than point mutations, such as structural variation, is going to be the next frontier, and really pulling that data and aggregating it in a meaningful way to make headway in therapies is very important, and we should focus on this in the future. Thank you so much, everybody.